In this video, we'll talk about the concept of tonicity. Now, let's start off by defining what it is. So tonicity is really a way of looking or comparing osmotic pressure gradients between two solutions. And in this case, we're talking about a cell and a cell membrane. So we're looking at the cytosol versus the extracellular fluid. Now, typically in body systems, we want to make sure that the extracellular fluid is isotonic. Now, what do we mean by isotonic? Think of iso. Iso refers to basically the same. So what this means is that both fluids have approximately the same concentration of solute. And in the case of isotonic, there's no net movement of water across a plasma membrane. So there's no volume changes in either compartment. In the previous video, we talked about the concept of osmosis, how with a semi-permeable membrane, water can move in order to equal out the solute concentration on both sides. So here's a case of isotonic. As I said, if you have a look at the picture here, you can see that there's the same concentration of solute outside the cell as inside the cell. So what does this mean? It means that at any given moment, there is basically the same amount of water moving out as moving in. And that means that there's going to be no net change to the water volume here or the water volume in the immediate area around the cell. And this is what we want, is isotonic. Now let's have a look at another case. This is hypertonic extracellular fluid. So let's have a look, see what we're looking at here. So here we have a cell, and the cell has a funny shape, and we'll talk about why it has a funny shape in a little bit. But you can see here the amount of solute is much greater outside the cell than inside the cell. And we know, based on the concept of osmosis, what's going to happen is that because the cell membrane is that semi-permeable membrane, water will exit the cell. And of course, as the water exits the cell will shrink and lose internal volume. So this is what happens if you took, say, a red blood cell and you put it in a hypertonic extracellular fluid. You will see that the osmotic pressure gradient pulls water out of the cell and the cell shrinks. There's a fancy name for this. It's called crenates, the shrinking of it. So you can imagine this would not be a good case if your red blood cells start to shrink. If all of a sudden you put, if your blood became a hypertonic solution. All right, so let's look at the third one, which is a hypotonic extracellular fluid. Hypotonic extracellular fluid. And in this case, let's have a look at the diagram. You can see outside the cell, the amount of solute, or the concentration of solute outside the cell in the ECF, is lower than inside the cell. So you would know, based on your knowledge, that water is going to rush into the cell. And of course, the cell is going to expand and expand, and eventually it may get to the point where it's actually going to lyse. So with hypotonic, okay, so hypotonic, right? What happens is the osmotic pressure gradient pulls the water into the cell. The cell is going to swell, and if it swells too much, it's possibly going to lyse, which means it's going to rupture. So these are three concepts we should be familiar with in three terms. And once again, that is what isotonic means, what hypertonic is, and hypotonic solutions and their effects on cell volumes. And once again, make sure you read your textbook. The textbook gives a really good overview of all this. It goes into detail, and it also has a number of great summary diagrams. So here you can see, this is from the textbook. You can see that they're just walking you through all three of the things we just discussed. So remember, you paid for the textbook. Use the textbook. It's fabulous, in conjunction with our notes, our lectures, and also these videos. Okay, I'm going to end it there because I just want to address the concepts of tonicity.